Pleasure to introduce the first speaker of this session, Professor Ruedi Aebersold, a pioneer in the field of proteomics who really needs no introduction, since I'm sure you all know him for his work on SWAP-MS and many others. Professor Aebersold is a Swiss-Canadian scientist who completed his studies at Caltech and is now a professor at ETH Zurich and the University of Zurich. He is the recipient of many numerous scientific awards and is on the scientific advisory board of a number of research organizations and has served as a senior editor for molecular and cellular proteomics and molecular systems biology. He was also the co-founder of the Institute for Systems Biology in Seattle and has also co-founded several companies. So please now join me in welcoming Professor Aebersold to the stage. Thank you.
Okay, good afternoon. It's a pleasure to be here. I would like to thank Max Quant organizers for the invitation to come here and to allow me to talk to you about an issue which I think is very important for science in general, for proteomics, and an issue that is also directly linked to what you're learning here, I think, in this course, namely to reproducibly generate research results or to generate reproducible research results. And as you will see, software and the way you use it is one of the key components of this, but it's by no means the only component. And now first I will start out with a disclaimer and then with an outline. So most of the data discussed here or show will be our DIA data, but I think that very similar results, certainly the results with similar properties, will be generated with DDA, MS, with or without isotope or isobaric tagging, as you learn how to do here. And I also think that the points that I'm making are more general. They're not dependent on a particular method, but they're general to the field of proteomics. So, there's an outline. First, I would like to discuss briefly why we should worry about proteomic results in terms of reproducibility, and I will make a political point and a scientific point. And then I will ask 3 questions and provide some data, some answers with data. And the questions are if 2 or more people have the same proteomic data. Now this will be DIA data, but it doesn't matter. I think the same question we could ask for DDA data of any kind. Can they generate the same results? If 2 or more people have the same sample in their hand, can they generate the same proteomic results? And if 2 or more people have to do the same experiment in a cell line, do they get the same results? So these are kind of important questions for the field, and I will then walk through one after the other. So now why should we even talk about reproducibility in the first place? And this is for many of you may not worry about that or may not follow this discussion, but it is, I think, an extremely intense discussion, and it's a dangerous discussion or dangerous situation for scientists who are in the middle or somewhere in a research career. So studies show that only a fraction of published science is reproducible. And this fraction is surprisingly low. So these are a number of studies that were done, so where scientists were specifically allocated, for instance, a company like Amgen, but also in other scenarios, to reproduce in their shop results that have appeared in a publication, not just any publication, but usually in high impact, high visibility journals. So these are the articles that people read and draw conclusions. And so this is one study where they could reproduce about 10%. This is another study where they could reproduce about 20%, about 30% of the published results. So on a practical level, this means A government like the US government, I think the EU would be similar in magnitude. They spend tens of billions of dollars, research dollars, or euros per year. And if only a fraction of the results of this work leads to results that are actually reproducible, therefore can be considered firm, then the politicians who might be, especially science-adverse politicians, might eventually say, well, a lot of this money is wasted. We as scientists would certainly disagree with this statement, and part of my talk will be to show that it is not so simple. I mean, in fact, it's a very complicated issue, this issue of reproducibility. So now, Nature Publishing Group should be commended on taking up this issue and actually not just say, well, it is a problem, but catalyzing a somewhat sophisticated discussion. And what they've done is they broke this big term of reproducibility down into various sections. Design of an experiment, reagents, data acquisition, statistics, validation. And you immediately see that when we talk about proteomics, terms of reagents, data acquisition, statistics, these are issues that affect us as proteomic scientists, certainly at these three levels, very intimately, and that's why it is important that we understand what the tools are doing, what is the statistical base, and how should these tools be used. So even within scientists, among scientists, there is a perception that there is a problem with reproducibility. This is a survey, again, from Nature Publishing Group. They surveyed approximately 1,500 scientists, and they asked, is there a reproducibility crisis? And you see that about maybe 80% or so of the scientists think there is at least a slight problem. This is a large number of scientists, of those who actually do the work, and so I think we need to take this seriously, because we are, of course, the ones, the scientists, who think we know what we're doing. We're not considering usually crisis as something we should maybe directly act on, or at least stop doing research. We would want to see a crisis and find a solution, as opposed to potentially some science-adverse politicians who might say, well, these guys don't know what they're doing, and basically say, let's give them funding. So why is this important? Reproducibility of results is the bedrock requirement of experimental science. If an experiment cannot be reproduced, we cannot trust it's the result. Now, as I'll show, the issue is much more complicated than just say it cannot be reproduced. There's issues of various levels, and I think we need to have we need to develop a differentiated picture. I think the political threat is that perceived or really poor reproducibility is construed, oftentimes with poor competency, or worse, as poor ethical standards. So it is oftentimes construed or equated, something not being reproducible, that one of the two people who tried to do the same experiment and arrived at different results, one of them didn't know what he was doing or was cheating. And I think distinctively this is not true. This is true probably in very, very few cases, but I will show that the situation is much more complicated, and that in vast majority of cases where something is not reproducible, that there is a very sound scientific basis for that, which we should of course understand and alleviate, but if it is not so, that poor reproducibility immediately means incompetence or cheating. So then there's of course scientific reasons. Reproducible analysis of sample cohort is the basis of data-driven research, which many of you who are learning how to do proteomics or applying proteomics are venturing into. We measure data, we generate results, and we infer something out of these results. This is referred to as data-driven. And I will come back to biomarker studies. We could also name systems biology studies, but these are broad areas where we need to deal with cohorts. It's not a question where we have a sample and we want to determine the inventory of proteins in that sample, but where we have a cohort of samples where, for instance, we would like to find a biomarker for cancer. For that, we need to have a cohort of control samples and a cohort of cases, and we would need to work with this data to learn something. For instance, finding a biomarker that differentiates these groups. And this is the first part. I want to now get into a little bit of data. So the question is, if we are using mass spectrometric tools to find biomarkers, for instance, to separate cases and controls, these are all individuals, some may have cancer, we'd like to find a marker that differentiates the two groups, then we will do frequently a proteomic experiment. And here Matthias Mann and I wrote a review about 2 years ago where we talk about the various options a proteomic scientists have in terms of a range of flavors of DDA methods, SRM, PRM, and data-independent analysis, DAA. And all these techniques can be used and are used to address this problem. So what we would like to ask is if we have such a problem that we would like to separate two groups, we have a cohort, we have a certain budget in our laboratory to do this study, where should we put our effort? Should we do more subjects or should we do more proteins? We can't do both at the same time, at least not with current methods, because going deep in high numbers of proteins per sample comes at a cost of fewer samples processed, because this normally means, and we go very deep down into the proteome, this usually means we have to somehow fractionate the sample into fractions, and then we spend more time and cost. So this is the topic I'd like to discuss now. And this is a study that we've been doing together with a statistician, Olga Witek, specifically a PhD student in Olga's lab, Ting Wang, who is doing this experiment or this simulation. It's a computer experiment. So what she did, she took a number of data sets from the literature. These are published papers. DDA data from CPTAC, SRM data, which were originally from our lab, and SWOT data, which are from our lab but not actually published. And she then made, from the results shown in these studies, she made a plot for each one of these data sets, where she has basically the intensity, the abundance of the protein that were measured in these studies on the x-axis, and then some form of variability, standard deviation, on the quantitative variance, so to speak, on the y-axis. So we see that when in these plots, for very highly expressed proteins, we have relatively little variability. If we go down in the abundance of the protein, then we get closer to signal-to-noise level, the variability goes up, and then this is an artifact I'm not going to discuss. This is an artifact of spectral counting, because on very low levels of spectral counting, then it becomes discrete. You either have 0, 1, or 2 spectral counts, and these clearly cannot be used as a confident quantitative indicator. So we basically can chop this off here, and we see that the lower the signal, the larger the variability. This is SRM data. This doesn't really happen there to the same extent, and the reason is because in SRM, the proteins that are being targeted for measurement are usually highly vetted for nice behavior, good confidence of measurements, low variability. Basically, these are good behaving proteins because so much effort is put in to develop these assays. This is DAA data, same deal. We see high abundance is quite low variability. Low abundance proteins is higher variability. So this is usually referred as trumpet plot, and is basically common to most methods. So what did Ting do? I skipped this. This just shows that from other techniques, we always see, to some extent, the same behavior. High abundance proteins. This is data, DIA data from a Lumos instrument. This is actually performs the nicest data we've seen so far. The proteins are measured very precisely with low variability, and then also it goes up when we get to signal to noise, but the effect is relatively moderate. Okay, so what did Ting do then? She learned basically this behavior from the published data, And she then segregates these proteins into 5 quintiles of expression level from the studies. High expressed, not quite high expressed, and so on. And then she puts in a computer, so she learns this behavior in a computer, and then she simulates into each one of these quintiles three biomarkers. These are synthetic biomarkers, nothing to do with measurement in the mass spectrometer, and the biomarker is defined as a protein that would have a 1.5-fold difference in abundance between case and control. So this is rather strict for proteomic standards, but that's what she did. And she sprinkled 3 biomarkers in the high level, high intense proteins, less high to the low abundance proteins, which have more variabilities. So up to here, just to give you an idea, up to here, the 3 quintiles would be about 1,000 proteins measured with 9 protein predictive markers sprinkled in, and here would be something like 3,000 proteins. Okay, then she took this and she separates these. She basically makes 2 groups, a training group and a validation group, and she does this by random forest and tries to simulate the predictive power of this data set after hundreds of simulations, you varying either the number of proteins that would be considered from 60 to about 5,000, or the number of biological replicates, which is basically the cohort size, from 10 to 400. And then she basically calculates how do these scenarios in the various background of protein measurements with DDA technique, SRM or DIA, how do they behave in terms of predicting correctly within the realm of this simulation whether a person is in a control group or would be a subject, for instance, with cancer. Okay, so this is the output. This is the accuracy of the prediction. This would be the number of correctly classified subjects, true positive and true negatives, divided through the number of subjects. So basically this determines how accurate someone who wants to separate this group would be if they were to use any one of the scenarios of numbers of proteins versus number of subjects. And we see that so the greener this is, the better. We see that we reach very high accuracy when we go up in the number of subjects. We also see something that is somewhat counterintuitive, that if you have a low number of subjects and you keep adding proteins, then eventually your predictive power goes down. And the reason for that is, one reason for that is that when we keep adding proteins that are measured by any of the proteomic methods, the results eventually become more noisy towards the low end, and then these noisy proteins will obscure truly achievable biomarkers, which would be observed if less noise is in the system. So the other objective is the sensitivity of this prediction. That's basically how often or what's the frequency that the same proteins which are true biomarkers are actually showing up among the top segregating proteins. And we see again that when we have few proteins, a small cohort of 10 to 40 samples, this is not robust in the sense that the sensitivity is low. So every time we would do this, different proteins are showing up as biomarkers, and the whole thing becomes only sensitive or robust when we have a fairly large number of subjects. And from these measurements, and we also see that for the sensitivity as well, the more proteins you add, the less predictable or the less sensitive it becomes. Again, the same issue of observation. So we can do this for all kinds of simulations. Ting has done that for different data sets on different instruments, different techniques, DDA mass spectrometry, SRM, DIA, PRM. And every time it's the same. It is the more subjects you add, the better. The more proteins you add is not necessarily the better, as long as you also introduce noise, And it also does not matter whether the biomarkers that are simulated in there are in the high expressed, middle expressed, or low expressed proteins. The numbers are different, of course, whether this is high, moderate, and low, whether the proteins are in the high, moderate, or low level of expression. But the trend is always the same. More samples always wins. More proteins is beneficial to some extent. And then it becomes counterproductive because we add noise. So what does this mean? We'll conclude this part and say this simulation experiment where Ting uses real behavior of noise and quantitative dependency on variability from real experiments, we learn that more proteins decreased predictive accuracy and sensitivity of identifying predictive proteins, especially if we add a lot of noise. More subjects increase the accuracy and sensitivity. Less technical variability increases performance. This is, of course, expected. And the conclusion would be that experimental techniques that we optimize for proteomic measurements should be optimized for throughput of biological samples and low technical variability and high reproducibility. That's our conclusion, where we should go with the proteomic techniques. Unfortunately, the new instruments that we have available are all pointing, of course, in that direction. So the landscape of proteomics is certainly moving into the right direction, including, as I will show just in a minute, also the software environment. So I now framed this simulation in the point of a biomarker study, but it is important to know that this applies, the same considerations apply for other data-driven approaches, for instance, clustering, machine learning, and so on. So it is not just focused or limited to biomarker studies. Okay, so now this was justifying the rationale from political reason and for scientific reasons why we should worry about achieving high degree of reproducibility in research results. Now I would like to answer the question, if 2 or more people have the same proteomic data, can they generate the same results? So this is now in this graph from Nature would be basically addressing this particular area here. And I think this is an important area, not just for proteomics, but of course for all fields of science. And in fact, when you look also through genomics, it is not so clear whether the genomics software tools that people use have been properly benchmarked compared to each other, and whether people can confidently say, if I use tool A or tool B, do I get the same result? So here we undertook a study to address this in the world of DIA, but I think similar things would be, of course, very useful in DDA world, although these types of analysis are actually quite tricky for technical reasons, because one needs to have some form of ground truth. Otherwise, it's not so that whoever discovers more proteins from a sample by using another search engine automatically has created a better result, because these identified proteins could be a lot of false positives. And I think that's why many of the leading software tools, including, of course, exemplary manner Max Quant, have very tight control of the false positive rates, have error control at the peptide as well as at the protein level. So here we generated a sample that consists of 3 proteomes. This is E. coli, yeast, and human. And we created 2 samples where these 3 proteomes were mixed together at different ratios. We know, of course, when we do mass spectrometry, whether a protein is from coli, yeast, or human based on the sequence. And we should then at least be able to say whether the proteins identified from these 3 species were the same or different from different software tools. And we can also see whether the quantitative ratios detected are actually accurate, because this is ground truth. So these samples were both run multiple times in various forms in a triple TOF mass spectrometer in DAA mode. And then there are 5 software tools that analyze this data. OpenSWOS, which is from us, SWOS 2.0 is from SciEx, Skyline, which is from Brendan McLean and Mike McCoss, Spectronaut from Biognosis, and DAA Umpire from Alexei Neshwisky. Each one of those has different features. These 4 here use libraries to search the data. This one does not, well, or more specifically, it generates a library on the fly. And the study was done by Pedro Navarro when he was a postdoc in our lab. He's now with Thermo. So then, this is basically the experimental design. We generated these samples. They were analyzed multiple times by different instruments. And then the data were sent to the developers of these 5 software tools. And they reported data back. And we have here an iteration 1 and an iteration 2. Turns out, after iteration 1, no one was super happy with the results. And then people got together. We had kind of a bug finding and correction meeting. And everyone got together in one place. We discussed these results. And then everyone did some improvements on the software tool. This was actually an impressive event because everyone who, from these 5 developing teams, they were willing to accept that there is improvement to be done in their tool. They got together, shared the ideas, and it kind of worked. And maybe it's best seen over here, where we see the protein. This would be the ratios expected for the various proteins from the various species, and these are the different tools. So the lowest one is the tool without. This is a DAA Umpire without a pre-cooked library, but this is a detail I think is not interesting for this course. So everyone, we have iteration 1 or iteration 2, and we can see probably graphically that everyone kind of improved a bit. This is also seen in this graph here where the ratios are plotted from the before the improvement and after improvement. This is the target value for the proteins in the least intense percentile. That's when it gets close to noise. So these are proteins down here. And we see that every tool improved a bit and gets quite close to the target value. Some actually hit it. So this means that irrespective of what tool someone would use to analyze such data, they come relatively close to the ground truth from the quantification point of view. And every tool experienced some improvement. And of course, that's kind of a nice thing to summarize. This is the identification. So overall, there were 4,700 proteins identified. And these are the typical Venn diagrams. Let's not look at peptides so much because it's many. So this is 3,950 and 500. They were seen by all software tools. About 4,500 of 4,700 were seen by 3 out of the 4 software tools. And it is similar for the inclusion of the DAA Umpire, which does not use an a priori library. And you see many zeros and very low numbers, 0, 1, 36, 26, and so on. Very low numbers of proteins that were only seen by one of the tools and not by any one of the other. This is certainly within the FDR range of the analysis. So I think what we can say is that this community effort in this DAA field resulted in a suite of software tools that we can quite confidently conclude that if someone is using any one of these software tools competently on good quality data, that they will generate results which would also be achieved, at least very close to those results would also be achieved if they used either another tool or if someone else used another tool on the same data. Okay, then I would like to go to the next point, and that is if 2 or more people have the same sample, can they generate the same proteomic results? This, of course, now goes a step further on this nature list, and this now addresses the issue of data analysis and acquisition. This is probably what we would like to achieve in the field of proteomics if we want to have reproducible methods, that it doesn't matter who does measurements and data analysis, provided that they're competent, they should get a similar results like someone else measures the same samples. Okay, let's see what happens here. We again generated a community effort. It's a cross-lab study that tests the reproducibility of results obtained in different laboratories by DIA measurements. We generated a sample which consists of HeLa, or HEC293 cell lysate, a human cell line, and then we spiked into these samples isotope-labeled peptides, in total 30 peptides, in different ratios and different conformations. So overall, we generated samples, 5 samples specifically, where these isotope-labeled peptides spanned 6 orders of magnitude in dynamic range, and each sample had a subset of these isotope-labeled peptides. And these samples then we sent to 11 participating laboratories. This is the geographic location. And each one of these labs agreed to run on day 1, day 3, and day 5 of a week, let's say Monday, Wednesday, Friday, these samples in a certain order. So they would inject these samples, 7 samples a day, 3 days a week. And then they reported the data back. And this allowed us, because the way this was designed, to see how reproducible is this within a day in the same laboratory, across a week is in the same laboratory, and across the laboratories. So we would like to know, do we detect consistent set of proteins across labs? And then what is the quantitative characteristics across labs? This is the result. This is a total of, I should say that everyone sent the data back to us. We used the same software tool to analyze all this data, either individually or in aggregate. So these are the sites. Each one reported 3 times 7, 21 files, and they were searched. So this ended up in the close to 5,000 proteins identified at an 1% protein FDR. And we can see that these labs are quite consistent. So there's some that did not quite as well. We see some where things kind of got worse. Maybe the instrument got dirty. These did incredibly well. But overall, everyone did quite well. We also see probably the most important thing, part of this graph, is this blue curve, because it's the number of accumulated proteins over eventually more than 230 runs of the same sample. So this is important, very, very important curve, because this means the reason that, the fact that this is a flat, reasonably flat line means that if we add here another 50 or 100 runs, that these will not find new proteins. And that means, basically, since we're constantly running the same sample under roughly same conditions, this is plausible, because why would someone all of a sudden find a lot new proteins when this has been done hundreds of times? So the fact that this line is actually flat means that the data analysis has the inflation of false positives in large data sets quite well under control. So this is actually an important curve which should be observed every time when very large data sets from dozens or hundreds of runs are being analyzed in aggregate. We would not like to see a curve which kind of keeps creeping up infinitely, because these are very likely false positives, provided, of course, that roughly the same sample is analyzed all the time. Okay, so this is another way to show the results. This is proteins listed here in a decreasing abundance, and we see that about 80% complete. That means the protein was missing in about 20% of cases. For 4,000 proteins, these proteins were observed in about 80% or more of the 230 runs. So this is, from proteomic point of view, quite a nice reproducibility. And of course, there is proteins down here to the noise level, which are not seen by all. And this would be probably proteins detected here, well, not detected by this group, but not by this group, because they simply did not observe the same number of proteins. So now another issue that came up in this study was that each group that ran this was basically considered a block. We see here behavior where some are very stable. This one is decreasing. This is simply protein abundance is shown, and this will be the decrease in performance of the instrument across this week. And then this is very easily rectified by mean transformation. Now so basically we're talking here about batch correction, which is an issue that will come up once we start running relatively large cohorts. There's various effects, various types of batch effects that we now learned exist and have to be corrected in different ways. This one here, like decreasing instrument performance, is relatively easily corrected by simply mean transformation, but other batch effects are not so easily detected and corrected. And here is the effect of batch correction on the quantitative ratios. These are dilution series from different laboratories achieved with the isotopic labeled peptides, and after normalization we see that each one also generates relatively nicely reproducible isotope dilution curves from the spiked isotope peptides. So I think what we can say is if people are careful with their instrumentation and they are careful experimenters, I think what we can claim now in the field of proteomics, that if 2 or more people have the same sample, they can generate maybe not identical but fairly reproducible protein results. Now I would like to come to the most complicated issue, and that is if 2 or more people do the same experiment in a cell line, do they get the same results? So this is now related to reagents, cells, who it could also be animals. For instance, mice, or yeast, or flies, or worms. But we did this analysis with cell lines because they're extremely widely used. These are human cell lines, and we chose HeLa for this analysis because HeLa is one of the most widely used cell lines, and as last count, there's almost 100,000 publications in PubMed of studies that used HeLa. So we of course assume that, and the number is of course increasing over time still. So of course we assume that if 2 people are taking HeLa cells in their laboratory, and they do an experiment perfectly the same steps like the person in the neighboring laboratory is doing, and they do everything perfectly, they do perfectly controlled measurement, they do perfectly controlled data analysis, they should get the same result. And the observation is that this is not so. And I explain now why. So the aim of the study was that we would do a cross-lab study addressing the biological reproducibility and the effects of the variability in the biological source material that many people use for experimentation. So we collected HeLa cells from a number of laboratories, 14, who gave the cells they use actively at the time in their lab, and we did multi-layered data measurement and analysis, and to understand how different these cells are at the level of their material makeup, or whether they are the same, and if they are different in their molecular makeup, whether this has an effect on the research results obtained from these cells. So this is the experimental setup. These are 14 cell lines that we obtained. They form in 2 large groups, Kyoto variant and CCL2 variant. This is not important, but these HeLa cell lines at some point in time split in the past. So there's kind of 2 flavors of them. And importantly, we had access to cells from a very careful experimenter, Wolf Hart at ETH, who freezes down his HeLa cells periodically after, let's say, 10 splits. He freezes an aliquot down. So we could get basically historical tract of how these cells changed, if at all, in Wolf's lab over time. Over about 40 splits. This is, just to put this in perspective, this is roughly the time a master student in Europe would take to complete his thesis. So the question would be, is this the same cell at the beginning or at the end of his master thesis? And then we did measurements, copy number variation, RNA expression, steady state protein, and protein turnover by pulse like labeling. This simply shows, this is a multidimensional analysis, to the reduction to show that these cells are actually rather close together to each other and different from other cells that are used. So they are not, it's not like people changed cells or made a mistake. These are still HeLa cells, but as we'll show, they evolved. They have different phenotypes. Some divide about every 17 to 20 hours. Some take almost 30 hours to divide. This is shown here by the accumulation, basically, of cells in a culture over time. So if you already have a phenotypic difference, they do not behave the same. In all these cells do not behave the same under the same conditions in terms of how quickly they grow. So this, I probably skip. This is just to show when we did this proteomic analysis that we get relatively good correlation between repeats, and we see this here, between whole cell process replicas, these are technical replicates, we achieve a Pearson correlation between correlating repeat 1 against repeat 2 of about 0.98 or slightly higher. Between cells of the same strain name, CCL2 or Kyoto, this is slightly lower, and between CCL2 and Kyoto is even lower, but still above 0.9, so this is a fairly high level in terms of technical reproducibility, but it shows that we actually do see different signals because the further apart the cells go from each other, the less the correlations. This is encouraging to see. Okay, now let's see at how the genome looks like. This is genotypic variability. These are the chromosomes of the cell, and normally every one of our cells is supposed to have 2 copies of each chromosome because we're diploid organisms, and this is not so for cancer cells like HeLa, also not so for many other cancer cell lines. And here we color code the absolute copy number from 1 to 5 or 6. So red means there's more than diploid, and the very dark green is less than diploid. We see that each cell, these are the cells listed here in these circles, each cell looks different. And so, for instance, this chromosome looks fairly similar between all, but here we have blocks of sizable chunks of a chromosome that in some cell lines is heavily multiplied, high copy number, whereas in others it is not multiplied as much. And here, this region is not multiplied in these cells, but it's in this one. So we can make all kinds of stories, but the cells are not genomically the same in the point of view of copy number variation. And now let's look at the 2 cells from Wolf Hart, which are about a master thesis apart in terms of culturing in the same laboratory. We see that this one here, this chromosome 15, was lost, was massively amplified in one, and was about normal in the other cell lines. And also we see here a change in ploidy copy numbers of an almost incomplete chromosome. So even within about 40 division or 40 splits of these cells, the cells are no longer the same from a genomic point of view. Now we might say, okay, this doesn't really matter, but what about, how does this affect the proteome and eventually function? So this is now data from 2 chromosomes, chromosome 11 and 1. These are the cell lines in this axis. And we have here the copy number variation, which we just looked at. We have mRNA abundance patterns and protein abundance patterns. And everyone can see immediately that what looks like very sharp islands of ploidy here get washed out and get further washed out at the protein level. So the more the information is going from the genome to transcriptome to proteome, the more complicated the effect of these ploidy changes becomes on the makeup of the cell. We also see areas here which are basically entirely green, which are roughly normal ploidy, And we see that in this green, at the level of the chromosomes, the genomic level, and we see that at the level of the mRNA, we see already red ones come in. So this means that even from areas which are not amplified, genomically, transcripts are upregulated. And the same is, of course, true for proteins. So it's clearly a very complicated relationship between copy number variation and the transcript profiles and the protein profiles, and they're clearly not easily predictable. These are simply correlations, Spearman correlation between the 14 HeLa cell lines copy number versus mRNA, mRNA against protein, and so on. We see that there's nowhere none of these levels is a very high correlation, and it's very broad, which means that various genes, various cell lines have different values. So it's not basically the essence of this slide is what happens at the genomic level does not have predictable consequences, neither on the transcript nor on the protein level. So this is, of course, not an interesting statement by itself. So we tried to come up with some rules, and one of the rules was, or explanations, what biology could be underlying this different percolation of copy number changes throughout the cell biology. So first of all, we see that here at the level of mRNA, to protein loss, basically degradation of proteins, the data deviation from the ploidy-induced ploidy changes detected in the cells is decreasing. So clearly what we see, the further away we go from the genome, where let's say a particular chromosomal locus is maybe multiplied from 2 copies to 5 copies, then we see that this amplitude is continuously buffered, increasingly buffered, the further we go away from the genome. So the mRNA, this is basically the deviation from the ploidy event induced. This is protein, this is K-loss, so we see a decrease in the amplification. So whatever signals the genome provides from genomic variation is tuned down, is buffered away at the protein level. We also show this, I don't want to discuss this because it takes a long time, but that various compartments of the cell have different behavior, how genomic variation is affecting the protein landscape in these organelles, for instance, the ribosome, centrosome, mitochondria, ER, and so on. But what I would like to now finish on, or get close to finish, is to say we see this buffering occurring. So clearly the cell has developed a mechanism that changes that are being induced from the genomic level are interpreted by the cell in a way that some of these amplitudes induced by the genome are buffered down so that the effect at the protein level is smaller than the genomic effect. So we tried to come up with a way to explain how this might be, and we did this based on statistical argument. And I am now showing what we try to maintain here. We assume that it's not just the protein, but it's the way the protein is organized in protein complexes, which provides a stabilizing entity to the cell, or to put it differently, a buffer of amplitude changes in the cells. So we segregated the proteins that we could detect, and for which we determined mRNA abundance, protein abundance, and degradation rates into 2 large groups. The one group is what we call complex in proteins, and these are proteins which we know that they occur in the cell in protein complexes. And the other are complex out proteins, and these are proteins which we do not know that they occur in a complex. They might, but there's no, as of now, no evidence. So some of these complex out proteins might also be in complexes, but we don't know, and so the difference we see between complex in and complex out in reality might actually be larger than we detect here. So then we take these proteins in these 2 groups, the pink and the green, and we do various forms of correlation. For instance, we correlate copy number, variation, and mRNA levels, and this gives for the complex in and complex out. The actual numbers here don't really matter, but the fact is that the important observation is that there's no difference how a particular copy number variation is interpreted at the level of the mRNA, whether a protein is in a complex or is not in a complex. This is kind of expected because how would an mRNA know whether eventually it has something to do with a protein complex? This is different when we go to copy number variation versus protein. We see clearly that complex in proteins are down buffered in their correlation compared to the complex out proteins. And we see the opposite when we go to copy number versus protein loss, that then the correlation is higher for the complex in protein, complex out proteins. So what we basically see here is that if this genome is unstable and creates additional copies of a particular locus, this leads in some convoluted form to higher number of transcripts, not directly proportional, but tendentially. It will lead to some extent higher number of proteins, but these proteins are buffered down when they are in complexes, and they're buffered down through degradation of excess protein that is being detected, being synthesized. So I should finish up here. I just want to show now in the last slide, basically, that we now talked about molecular makeup of these cells, and we show that these cells which are used in experimentation in many laboratories are actually not the same source, and we can document this in the level of measurement of specific types of molecules. And now, but we could say this may be irrelevant because the cells will still do the same thing. So now we observed that when we treated these cells with a microRNA, this goes back to research that's been done by Wolf Hart's lab, which I don't want to explain, but L17 is a microRNA that modulates certain biochemical functions in the cell. And then we treated all these cells with this microRNA, and we clustered the proteins, the cells across the response now, and we see that the cells are not only different in their steady state makeup, they also react differently to a challenge. And this challenge is a challenge that is relevant for infection biology, and because it was observed that if these cells are infected with, for instance, salmonella, that this attenuates the expression of this microRNA. So we undertook an experiment to say, let's inhibit this attenuation by stimulating the microRNA L17 by a mimic for L17, and let's then infect the cell. So we basically prevent the cell from reacting to the downregulation of L17, which normally happens from this as a consequence of this infection. And we simply asked, does this, when we inhibit the ability of the cell to react with L17 to infection, does this have an effect on the infectivity of the cell with the salmonella? And it does. So we basically look at this graph here, where we see the second panel is the downregulation of infectivity mediated by abolishing effectively the normal response of the cell to the infection. And we see that every cell reacts differently. This cell doesn't react at all, but every cell reacts differently to this challenge. So what we can conclude is that when people are doing research with cell lines, this case HeLa, but I think this also extends to other cell lines that are cultivated in the laboratory for a long time, that they are not doing the same experiment in the same laboratory over time, and they certainly do not the same experiment entirely in different laboratories. So we see that the proteome organization is a major buffer of expression variability. We see that the proteome variability is connected to phenotypic variability, so the cells are not just different, they also do different things. And we think this has implications for experimental biology with using genomically unstable cell lines, because we can clearly demonstrate that if two people did the same experiment with different cells, which are actually variants of the cell, and they do not receive the same results, that neither one made a mistake necessarily, but simply they did a different experiment. And so with that, I would like to finish and state that I think, in summary, that this issue of reproducibility in research results is a politically hot topic, and I think we should make as scientists an effort to go out and talk to people and say, look, biology is really complicated. It's not that people don't know what they're doing, but there's underlying variabilities which we don't yet fully understand, and that may lead to irreproducible results, but it has likely a technical or biological reason which eventually will be understood. It's not sloppiness, and it is certainly in most cases not cheating. So with that, I would like to acknowledge my lab's contribution, or people's contribution from my lab whose work I talked about. This SWOS technique was developed by Ludovic Schia and Pedro Navarro with collaboration with scientists from AB SciEx. The software we use is from Hannes Röst, George Rosenberg, and Pedro. The HeLa study is the work from Yansheng Lu, and we got particular help from Wolf Hart in phenotypic measurements. The CrossLab study is the work of Ben Collins with Christy Hunter from SciEx with Yansheng Lu, and the CrossLab software benchmarking was basically the initiative of Pedro Navarro, and he then moved to the lab of Stefan Tänzer at the University of Bonn, where he completed this work. So with that, I would like to thank you for your attention, and I wish you a great week, learning a lot about proteomics, and I wish you best success with your research.
Thank you, Professor. We can now have maybe one or two questions.
Yeah, so we got some conclusions from you here regarding why it's difficult to reproduce biological data. Also, I wonder, what do you think about the case that we have this publish or perish scenario in science in general? How that adds into the low reproducibility?
Yes, so I think that's also certainly a factor, but I think this is actually the minority of cases. I mean, this would imply that people are not very careful and they make mistakes or make shortcuts. I actually believe that this is a vast minority of cases, and the vast majority there is reasons which eventually could be found out why things are hard to reproduce. For instance, like reagents are not the same. I mean, people here work probably not a whole lot with antibodies, because this is a mass spectrometry-based course, but it's well known that if you order some antibodies against an antigen and you do a Western blot or immunoassay, that not every antibody from every company that is supposedly hitting the same target will give you the same result. So I think these research reagents, methods, instrumentation, they should be somewhat benchmarked and try to show some examples of that. And I try to show with this cell line example that this may be very, very hard to actually achieve. We're actually trying to come up with some community discussion with guidelines how one could not alleviate this problem of unstable cell lines, but at least document that it is happening and document the state of the cells that were used in a particular study.
Thank you very much for this wonderful talk. I have a question about the proteomic, or what you call the buffering at the protein level that you see with the HeLa. Do you think this is something that is particular for cells in culture, or do you think this could be applied for any system or any cell type?
So this genomic instability is certainly very characteristic for cells in culture, and even not all cells in culture. This is particularly characteristic for cancer cells that are in culture. It has, for instance, been measured that, for instance, organoids, which are also eventually cells in culture, or induced pluripotent stem cells, can be cultured over a much longer time without undergoing this instability, genomic instability. There is suggestions from, for instance, Tony Hyman in Dresden to say one should abandon or ban basically all experimentation with genomically unstable cell lines. This is, of course, at some level a very good argument, because when we know it's unstable, why use it? On the other hand, we see there's 100,000 people at least, usually groups, have worked on at some level for years on some results, and I think it would be very tragic to basically say all this research means nothing. So I think one should find different ways. But it also applies incidentally to mice or yeast, where the strains are not frequently very well documented. And different strains clearly behave differently.
Yeah, but my question is, this genome instability that you see in this particular example with the HeLa cells actually is buffered at the protein level, right? You just see. Do you think this is also applying to any other system?
Yes.
Yes. This was my question. Thank you.
That's true.
Also following up on the same question of the proteome buffering, could it be just due to the fact that we collapsed then our result with proteomics into protein groups, and then in like complexes, you could think that one protein has a homologue and then it's substituted by a homologue.
Okay. Yeah. So this is a good point, but in this case, this is not actually what's happening, because we only use for all these analyses only prototypic peptides. So there's no protein groups. It could be, of course, that there's inadvertently protein groups, for instance, that we don't recognize. For instance, splice forms of a protein where we don't know that different splice forms exist, but in the classical protein groups where you have like isoforms from different loci which have some different peptides and some shared peptides, this is ruled out here.
So again, it's building on kind of the previous discussions. So this protein buffering is kind of really nice to see, but also kind of scary in a way, because if there is indeed a large amount of protein buffering in a case where there is so much genomic differences, how would you argue for using proteomics approaches for doing biomarker discovery? Because even very large changes in the genome might not lead to a proteomic change. And then the next part of that is, of course, then what is the phenotype of the cell? Is that being driven by the changes in the proteome, or is that being driven by the changes in the genome?
Okay, maybe to the last part first. I distinctively believe that the further away you are from the function. So I believe that what we see as phenotypes, as biomarkers or phenotypic indicators like whatever that is, tumor, faster growth, slower growth, whatever, is always rooted in some biochemical reason. So we should be able to say that if what part of the biochemistry of the cell is changing to lead to a phenotype. That, for me, is the bedrock of biological phenotypic explanation. So then if out of that follows that the closer you are to the biochemical function, the closer you should be to the phenotype. And that is, of course, with those molecules that actually carry out biochemical functions. And these are actually not proteins, but usually protein modules. There's very few proteins which carry out their function by just somehow floating around. Usually complexes are formed, ribosomes, spliceosome, proteasome. These are not individual proteins, but these are machines composed of multiple subunits. Sometimes they also contain RNA species. And what we do know from many, many cases now, where we also did measurements in flies, we did measurements in humans with ploidy changes, we did measurements in mouse over cohorts of mouse strains. Whenever we correlate either the genomic variation or proteomic variation to a phenotype, the proteomic variation and proteomic pattern is always closer to the phenotype. So I think this is a tremendous opportunity for proteomics, because we can claim we're closer inherently to the phenotype, and I think we should at least complement what genomics community is doing, because it clearly provides additional information.
So in the interest of time, let's thank Professor Aebersold again.